Hi, everyone, and welcome. I'm Monica Daly, and I just want to welcome everybody to our next episode in our series of workshops on integrative organismal modeling of movement. These workshops have been sponsored by the National Science Foundation Division of Integrative Organismal Systems to help generate discussion and collaboration between experimental and theoretical scientists to help the community make more effective use of analytical tools for understanding organismal biology. And these workshops will be recorded and shared as an online resource hosted through the UCI Center for Integrative Movement Sciences. And you can find that a link to that website on the workshop flyer. The workshop today is on using physical models in evolutionary biology. And with that, I will just go ahead and pass things over to Em Standen and the rest of today's speakers. Take it away, Em.
Great, thank you. I'm just going to get my laser pointer out. All right, so thanks for that introduction. Yeah, today we're going to talk about using physical models to understand biology. And I'm Em Standen, and I'm at the University of Ottawa in Canada. And our team consists of George Lauder at Harvard and Talia Moore at the University of Michigan, Henry Astley at the University of Akron, and Brooke Fleming at New Jersey Institute of Technology. And we are a group of engineers and biologists. And what brings us all together is our interest in biomechanics and using biorobotics and physical models to understand biomechanics in biological systems. Okay, so today's workshop is going to have 5 parts. We're going to start with an introduction, which is very simply why use physical models. And that's going to move into some really cool examples of physical models that come out of George Lauder's lab. And Brooke's going to give us a rundown of the really critical considerations that you need to take before building physical models. Talia is going to give us a rundown of 3D printing approaches. There are many and they have pros and cons. And so that's a great segue into how to make your models. And then we're going to wrap up with a lovely start to finish example using a physical model to test real biological hypotheses. And Henry Astley is going to be doing that for us. All right, so I will start with why physical models. And we want to use physical models because biology is complex. When we ask something about a biological system, we need to simplify because a whole organism, if you think about a tuna here, has a whole bunch of physiological complexity going on that if our question is about how it uses its tail, we can't extricate that physiological complexity from the functional complexity of the tail. And so physical models can be very simple or they can be complex, but what they do is they help us modify and compare differences between morphologies. So before you even start with a physical model, you need to know what your question is. And it might be, how does the tail fin work in a fish? Might be, how do mammals chew when they're eating? And it could be, how does a bird hang on to a perch in a windy storm? All of these things are important questions and they're interesting questions. You may have gathered them from watching YouTube videos or just being outside. The critical thing about biological organisms is that they don't do what you ask them to do on command. And so sometimes it can be really difficult to get your animals to swim at the range of speeds, let's say, that you want to test, or, you know, behave in any number of different ways. And so that's where physical models become really useful, where you can isolate the variables you're interested in and then test a range of behaviors or kinematics without having to get into the psychology of an organism, a living biological organism. Okay, so why use physical models? The first we just talked about is control, that mechanical and robotic devices allow precise control of the variation that you're interested in. You can change and tweak things really easily, unlike in biological organisms. The second is landscape, and what we mean is performance landscape, and I'm going to, in a couple of slides, explain what that is. But what this does is allows us to understand whether the organisms we're interested in are operating at a peak performance level or if they're constrained in some way. And I will get to that. And then finally, physical models can actually give us some information about cost and efficiency of biomechanical performance. And this is because we can change the morphologies and change the kinematics and see how much energy it takes to do so. All right, so quick example, the control aspect. Say we're just interested in how tail shape influences function in swimming. We want to compare a tuna-shaped tail to a bluegill-shaped tail. Well, there's a lot of differences between those animals that we can't control for. You know, muscle mass, life history, even developmental stage can be a problem. So what we need to do is extricate the one variable we're interested in from the complexity of the biological system. And so physical models allow us to do that. In terms of performance landscape, what we mean by performance landscape is if you look at this figure here, you've got a couple of variables you're interested in: speed on one axis, amplitude of motion on another axis. And with a physical model, you can actually make many different comparisons at different speeds and different amplitudes to create, in measuring efficiency, create this landscape of efficiency depending on the speed and amplitude you've got. Then when you go back to your organism and you look at what that organism performs at, you can see, is it landing at one of these high peaks of efficiency, or is it somewhere else on the curve? And what is the constraint that's allowing it to be at that curve? So looking at that performance landscape, measuring where your animal is, allows you to get deeper into more critical questions about why animals are not operating at a potential peak. So at this point, I'd like to jump into showing some examples of physical models that are coming out of George Lauder's lab. Now, George is really disappointed that he can't be here, but for circumstances out of his control, he was not able to be here. So we will miss him, but I will go through his slides and talk about some of the really cool stuff that's going on in his lab. So first of all, we want to talk about how there's a diversity of physical models that are potentially out there. You can have simple physical models like flexible plastic foils. You can have intermediate and then really complicated biomimetic designs. The great thing about the biomimetic design is that they accurately mimic fish shape and fin shape. They can do natural fin motions. You have big parameter spaces that you can access with these kinds of robots. You can alter the stiffness of the material that you make these robots out of, and you can measure the forces they're producing. The one major drawback is that they're very hard to build and really hard to control. So there's complexity there. On the other hand, really simple physical models are pretty interesting. You can alter aspect ratio of the flapping foil. You can change its material properties by using different materials. You can change the shape of it, which will change its wake structure. And they're very easy to control. This is on an oscillating, flapper. The major drawback, of course, is that they're not as biologically realistic. But I'm going to show you how even these very simple models can give us good information and fine-tune our questions. So on the bottom of this slide, right here by my pointer, is the outline of a trout swimming. And you can see that when we flap a foil in a flow tank, you get a really similar traveling wave that mimics the fish in a pretty good way. The great thing about the foil that you can't do to the fish is that you can change the length of the foil, you can change its shape, you can change its stiffness. And by doing all of those things, you can measure the forces and torques and get an efficiency for each of those behaviors, which is pretty cool. The other thing you can do is augment this a little bit more. And George with his students have done this in his lab. You can see here on the left are a series of different fish with fins in different locations. And if your question were to be, how is fin placement affecting force production or efficiency of foil flapping? You can change the position of fins on a very simple geometric structure to get an idea of how efficiency is changing based on fin placement. So really nice examples of very simple physical models that help us gain information about different shapes and morphologies in fishes. So we're going to walk through a very simple experiment. And the question is, how does the length of the swimming object impact its locomotor performance? And so if you think about it in terms of fish, it's very difficult to do on live fish because a small fish and a big fish, sometimes it's hard to get a good size range. Also, there are other developmental and physiological parameters that can change, as well as morphological ones, that can change between small and large fish of the same species. So using a flapping foil gets at exactly that variable, length, and how it affects locomotor performance. So if we put a flapper, and this is George and his students did this, put a foil onto the flapper and formulate a hypothesis. If it was a really, really short foil, they hypothesized that it would not have a very high self-propelled speed. If it was a really long foil, they also predicted that it would have quite a low self-propelled speed due to excess drag. And so the prediction is that at some optimal length, you're going to get an optimal speed output. And this would be the relationship. Now, when they actually did the work and measured at all different lengths of foil, what they found was actually two peaks coming out. So two narrow peaks with a trough in the middle. Very different outcome from what would be expected, and really interesting to think about from a physical viewpoint and how that relates to length of animals in biological systems. So very interesting. Another example of a really interesting use of physical models coming out of George's lab is his work on shark skin function. So sharks have really interesting skin. In the background of this slide is a close-up of the scales of a shark. So very complex. And they were interested in how that complex morphology might affect swimming performance. And so this is a really cool example of taking biological material and combining it with the flapping foil to get more data. And so they took a shark and actually took the skin of the shark and put it onto a flexible foil and flapped it. And as they flapped it, they measured the hydrodynamic forces. So this is a flapping foil in a laser light sheet. And they compared the foil covered in skin with a plain foil and understood differences in the hydrodynamic performance of the shark skin. What's really interesting is that sharks have different shape scales on different parts of their body. So a question that comes up is, what is the effect of scale shape on performance, hydrodynamic performance? And the way to get at this is to take those scale shapes, CT scan them so that you get a 3D representation of those scales, and then you can alter them. You can change their shape, you can change their positioning. And if you 3D print them onto a flexible foil, what you end up with is essentially synthetic shark skin that you can manipulate the scale's position, their shape, and understand how those attributes of the scale are affecting hydrodynamic performance of the animal. So here is an example of that. This is the foil with the synthetic skin on it, swimming in the flow tunnel. If you measure the forces that the flapper is experiencing while it flaps, you calculate the self-propelled swimming speed, then you can get a cost of transport for the foil with scales versus a smooth foil. So very cool use of a physical model. Of course, physical models can also be quite complicated. And this is another example coming out of George's lab, which is the tuna bot, and this is version 1.0. You can see here's a tuna up here, very complicated fin structures, but this model is very cool because it's taking a rotational motion and turning it into a flapping motion. And if you want to know more about actuation, please come to our workshop next Friday, and we'll be talking more about how to actuate your physical models. But in this tuna bot version 1.0, the goal was to create an autonomous robot that can swim and emulates the shape of the tuna tail. And so here are some examples of it swimming in the flow tunnel. You can see it's pretty effective. This picture down here is swimming in a light sheet, so you can see the hydrodynamic forces behind the fin of the fish. And then using these models, you can swim them at different speeds, see how much energy they produce or use, sorry, and get an estimate for efficiency of swimming given a particular shape. Now, of course, robots evolve. And so here's TunaBot version 2.0, and you can see, I think we can, yep, there we go. You can see that some additions have been made. You can see more articulation, flexibility along the body, more complex and biologically relevant fin structure. And what this robot allows them to do in the Lauder lab is really start to tweak and change these variables in swimming to understand what is it in the biological systems that are allowing these animals to be as efficient as they are. Okay, now hopefully that has whet your appetite for physical models. I'm going to pass the baton to Brooke now, and she's going to give you an overview of, before you jump into making your model, the considerations you need to make to ensure it's going to do what you want it to do. There you go, Brooke.
Thanks, Em. Thank you.
You're welcome.
All right, you need to drive the slides now. All right, so at this point, I'd like to invoke the phrase robotic model organisms, because in reality, that's what you're really doing here. You're creating a biological proxy that you can use in order to do repeatable testing. And this is something that you could share and other people could use. So that really is a great sort of part of the accessibility of using robotic model organisms or robotics in comparative biology. And so why would you want to use an RMO in the first place? Well, you might want to simplify really complex morphology because maybe your question is only looking at things like shape versus stiffness. You might want to simplify a really complex behavior. If you have organisms with multiple appendages and you're trying to start out with only looking at one or two of those, it might be easier to deal with that in a simplified model context. You want to be able to directly measure forces or energetics. And this is something that can be really complicated to do with living organisms, especially for those of us who work in the fluid environment. It's almost impossible to directly measure forces from a living organism. So by having some sort of structure attached to something that can read your forces, you can get a lot more information out of these organisms. Maybe your question is about animals that don't exist anymore. Maybe they're fossils, or maybe they're evolutionary missing links, and you're trying to understand an evolutionary process. Having a design that you can iteratively manipulate in order to address these questions can be really, really useful. And lastly, anybody who works with biological organisms know they'll do whatever they want. And so if you really want absolutely precise, repeatable behavior, having something that can demonstratively do that same thing over and over again can be a really useful tool. Now, so what are the considerations for building a useful robot? Well, to start, you have the organism, and you know that this organism exists in some sort of environment, which has a certain set of external factors that don't change over space and time and have been the same since the beginning of time, right? So normally when people think about organisms, they might say, oh, in what way is this organism optimized, right? So optimization is something that comes up all the time. But the problem is nothing in biology is optimized. It's not. It's because of evolution, right? So if we're going to think about what the organism is doing and why it's doing it that way, we really have to take into account that there is some sort of hereditary deck that this thing has been dealt, and those are the cards it has available to play with. And so the phylogenetic impacts include things like its morphology. It includes how that morphology allows it to perform, and that performance is ultimately going to dictate the fitness and why we see that particular organism that we do today. So knowing that, we can consider those things when we actually go to build a robotic model organism, and we build it for what it is and not for what we hope it would be, right? So we're really trying to understand the organism and create a system that we can use to then explore the actual organism and not an optimized version of it. That will come later. So we need to understand things like the material properties of the biological structures within that organism and the kinematics of how the organism moves. we can use those things together in order to validate our robotic system. And I'll go into that in a little bit of detail. And once we have our validated robotic model organism, then we can use that to explore a broader parameter space and ask those questions that we couldn't ask with just the organism themselves. This might allow us to test hypotheses with things like selective pressures and performance trade-offs in evolutionary history, and also ultimately allow us to build a more optimized design if we have a goal for a deliverable robot that is some way does things better than the organism itself. So even with all the organisms you can consider through history and with current diversity, there are still rules that are followed, not only in organisms, but also in robotics. So the first one is, as I said, those environmental physics are fixed. And so your robot and your organism both need to be able to perform within the constraints of the environment in which they are. And this is where using a actual physical model is nicer than, say, doing something in a theoretical computational sense, because you don't get to mess with the variables of the physics when you're dealing with a real, right in front of you model. The other thing is standard building materials. So bone is fairly consistent among all vertebrates, and it has, certain properties that we know that we can expect to be the case in dealing with them in terms of their stiffness, and how that's going to bend or not and react to forces that are applied to it in the external environment. And so keeping that in mind is important. There is some constraint to the way that organisms are organized. And this is important when considering things like where are the limbs relative to the center of mass? Because not only is that important for a live animal to move, it's going to be important for your robotic organism to move appropriately. And the last thing is, in evolution, it only has to perform good enough to survive. And so again, this is remembering that optimization is not what's happening here. You're really evaluating the system as it is and making a working functional model that does that thing good enough. And so by considering all of these, you can build a useful model that will be able to accurately represent the organismal behavior that you're interested in. All right, so the most important thing out of all of this is to ask yourself, what is your question ultimately that is driving your need to build a robotic model? So once you know your question, that will help you decide the level of complexity that you need for your particular question. It will help you determine the level of accuracy you need in replicating the biological system. And it will lead you down the path that you need in order to validate your robot to know that you can then use it for parameter space exploration. All right, and remember, I can't say this enough, even though I'm calling it a robotic model organism, it doesn't have to be the whole organism. We're not going for biomimetic here. We're going for an accurate representation of the physical system in the physical environment that allows you to answer your questions. So it can be a really simple model like this, where we were just trying to tease apart the effect of shape versus stiffness. And even having a series of really simple models, you can make broad comparisons by testing these, say, with fluid dynamics in our case. We were able to look at the more subtle effects of changes in shape and stiffness that's hard to do once you start introducing organisms that might be very variable in their morphology and have a lot of other differences that you don't want to include in your analysis. But simple might not always be the way to go if you have a really complex question. Another thing that we've been working on is looking at flying fish and their ability to take off from the surface while the anterior half of their body is in the air and only their tail is in the water, and how they're able to do this repeatedly. In order to do this, it's going to take a slightly more complex model, because we need to have a model that's able to deploy fins and to have powered thrust from the tail and have the right camber of the pectoral fins. So this is a work in progress. These things take some time, but getting the model right is important to be able to get all of the data that you want out of it. All right, so when you're trying to determine how accurate your morphology and your material properties and such need to be in building your robot, you have to sort of break it down to what's going to matter the most. And one of the big things is scaling. So there is going to be a reasonable limit to the size at which you are building your model. Whether you are studying a mouse or a giraffe, you might want to have some sort of limits into what you're going to be able to build. That also relates to the limits of your testing equipment and what you can reasonably test in the physical environment that you have available to you. In addition, there's going to be the laws of scaling of the physics of the system that are going to have some effect on what is the size of the thing you can build, and what are the speeds at which it can move, and what are the forces that you're going to be trying to measure. So considering all those ahead of the game allow you to not have to go back and change things as much after you've already started building. The material properties of your robot are going to be really important. And again, those are going to come from biological qualities. I like to call these the 4 S's. So the first S is the shape of your organism. And so imagine that you are doing research on fish and you could say, well, this is a fish, and then you have some fins on it. That's good. And then so, this fish has fins, and there might be a fish that looks like this, but odds are that most fish actually look like this or like this. And so these are all really important shapes if that's the sort of organism you're studying, and you need to incorporate that actual shape into your robotic model. When you are then, the next thing is stiffness. And so when you have a model or an organism interacting with the environment around it, the way that it yields or doesn't when in contact with that environment is going to be important in the forces it produces. And you can get a lot of the stiffness information that you need in many cases from things like CT scans. It can be really helpful. So the CT scan of this fin right here not only tells me things like the overall area of the fin and the length of the fin rays. If I were to cross-section through it, I can also get things like cross-sectional area and second moment of inertia measurements in order to be able to come up with some calculated stiffness that I can then try to use and replicate in my robotic model. You can also do material testing on biological tissue in order to determine what sort of material properties that you're dealing with. The third one is strength. You want to make sure that when you are deciding how you are going to build your model, that it is not too brittle or absurdly strong so that it doesn't react appropriately to the environment around it. And lastly, the surface that you're going to be interacting with. If you are having something that has to move across a substrate that is smooth and hydrophobic versus something that is, granular and gives way or has large obstacles in it, these are going to be very different things that your robot is going to have to contend with. So what matters the most here? You have in kinematics, hopefully you've been able to at least observe the animal actually moving, if not actually have kinematic data. But again, you can't build a robot based off of one optima because animals are not optimized. They're going to do a lot of different things, some well, some not. And you want to make sure that you are really understanding the way that this organism moves in order to be able to have a baseline for what the behaviors you are trying to produce in your robot. So again, there's this performance range, this performance landscape. So some of the variables that you want to take into account here are things like where is that center of mass? Where are the limbs located relative to that? What is the range of motion in the way that it moves? And, are there relationships among different components of the body when they're moving? And lastly, validating your model. So you've gone ahead and built a robotic model, but before you can test it as a proxy for an actual organism, there needs to be a validation process. And so we've gone ahead, say, for example, and we've used the CT scans of fish fins in order to get material properties like stiffness and length and area, and now have produced a good representative of this fish tail using 3D printed components. And we're able to actuate it, and we can look at the kinematics of our robot versus the fish and say these match up pretty well. It looks like we've done a good job matching these. And then we can actually look at the fluid dynamics of both the fish and the robot performing and say, yeah, we're getting the same amount of expected force generation here, the same amount of momentum imparted to the water, things look relatively similar. This is a good approximation of how this tail actually moves in a live animal. Now we can go forward and we can start to expand our experimental parameter space. All right, now in order to get these information, earlier in some of these workshops, a lot of people have covered getting CT information, so I'm not going to go into that here, but just to remind you, MorphoSource is a great place to download CT scans if you need access to them. And once you have CT scans, you can manipulate them in other free software, such as SlicerMorph is also a great tool to use, and they have a wonderful user community for bouncing questions off of if you need help. And if you're an engineer watching this, I strongly urge you to make friends with a biologist, because we would be happy to talk nonstop to you about our organisms, and you could get all of the biological information that you need. Now, if you need to design simplified parts and you're not going to directly 3D print anything from a CT scan, one of the things you can use is another free software is Tinkercad. And that is made by Autodesk, and it's really simple to use. My students use it in our bio-inspired robotics course, and they pick it up fairly quickly. You can basically pull in shapes and stick them together and manipulate them in order to make the raw geometries that you might want to 3D print for your parts. Then once you go ahead and you 3D print things, which Talia is going to get into in more detail soon, you can make them, say for here, we were making different stiffness fin rays. And so we could very easily manipulate the same piece over and over again in slightly different ways in order to have testable components. And one of the ways that we've used this specific method recently in my lab is in looking at the adhesive discs of Remoras. And so we were easily able to make a 3D model where we could remove individual lamellae. Those are the bars with the little spiky teeth on them that the Remora uses to help generate friction when it's attaching to a host animal. And so these allowed us to be able to answer our research questions about why is it so important to have a certain number of lamellae in order to have a certain amount of adhesive strength. And just as a little bit of a case study about that, so what we're really interested in is there's 8 species of Remoras, 2 of which are generalists and the other which tend to be more host specific. However, there isn't anything really different in disc morphology at all that suggests that they would have any sort of host specificity. But the only trend that you do see is that through the phylogeny, there seems to be an increase in the number of lamellae. And so we wondered if that was something that was selected for performance. So my graduate student, Kaylin Gamble, she made this disc that is 3D printed entirely. And what's really cool about this, is that this is a completely passive model that requires absolutely no mechanical actuation. It relies only on the underlying physics of the system. And I want to throw this in here because this is a really important consideration when you are doing an evolutionary project. If you include so many mechanical components that you can kind of overdrive the physics or override the physics of the system, then it gets hard to really understand the question in a comparative context about what might work better than something else. So if you are using an actuated system, you want to make sure that you're really cognizant about like how much energy are you putting in, because the motor is going to work no matter what you ask it to do. It just might take more power. So in being able to simplify this question, we wanted a system that we would be able to remove all of those variables. And also, we wanted to test it on biologically accurate surfaces. So we manufactured a number of surfaces that were representative of some of the hosts of Remoras. And in the end, our model was able to have a 50% greater adhesive force than we were able to measure in live Remoras, which is really cool considering the simplicity of the design overall. So finally, when you're using robotics as a tool in biology, don't forget about the biology part. The biology is key. You need to include the morphology and the material properties, the kinematics. The phylogenetic context could be important in understanding why it's so wonky in the way it does it. These all come together for the whole organism dynamics. You want to make sure you're keeping a similar environmental media and the physics are constant for your robot, so you have the right organismal environmental interactions that you would for the organism. And lastly, validate. And oh yeah, so always take home from Jurassic Park. You know, make sure that you don't over robot your robots. Scientists are so preoccupied whether they could, they didn't stop to think if they should. All right.
All right, thank you, Brooke. Sorry to rush you on some of those slides. All right, next we have Talia, who's going to give us the ins and outs of 3D printing. Take it away, Talia.
Thanks, Em. Yeah, so I'm really excited to nerd out about 3D printing. I have a lot of content on these slides, and I'm not going to talk about all of the different points on the slides, but the slides have the content so you can like screenshot them and think about them later. If you have specific questions about these, or if you want to brainstorm, or you think it'd be fun, or you just want to nerd out about 3D printing, this is my contact information. And I just want to say that I had a fantastic undergrad, Maggie Kohler, who's now a design engineer at Pratt Miller, and she helped me make a lot of these slides. Okay, next slide. Thanks. So the learning goals for my specific portion is I really want you to become familiar with the different types of 3D printers that are out there and be able to choose the appropriate technology for your application. I also want you to have access to some of the best practices for future work of your own and be aware of some of the common pitfalls. Okay, so yeah, here's kind of an outline of the topics. First, I'm going to talk about 3D printing terminology and different types of technologies. And then I'm going to get into a little bit of the details specifically for FDM, which I'll explain later. And those will be like slicer software supports and trade-offs and best practices. Okay, so 3D printing terms in tech. Here's the 3 different general categories that I came up with for 3D printer technologies. And so I know that they can be categorized in different ways, but this is how I'm organizing it today. So on the left, we have just adding material. So there isn't any material kind of in your print space, you're adding it to create your 3D printed object. And that'll include FDM and Polyjet. In the middle, we've got hardening resin. So we've got a liquid resin and we are using some kind of light to harden that resin into your 3D printed object. And then the third type you'll see on the right is melting powder. So you'll have powder made out of some kind of material. And again, you'll be using a laser to melt that powder and then let it cool back into a solid. Okay. So yeah, the first type of 3D printing I want to talk about is the one that's most common, easiest to get into. If you want to test out 3D printing, I would actually first suggest you talk to someone who has a 3D printer, see if you can print something on their machine. But then if you want to test out buying something, this is the one that I would suggest you buy. A lot of my students have actually bought these or built these and put them in their dorm rooms so they can complete projects at home. So fused deposition modeling, you have, it's also called like a fused filament 3D printing, because there is like a long filament of solid material that it's basically like a glorified glue gun on rails. And so it melts that filament and then deposits it into the 3-dimensional structure. This can be really, really cheap, and there can be multiple materials that you can use. So PLA, ABS, you can do lots of different types of material property materials. So some being flexible, some being dissolvable. There are also some printers out there where you can print with multiple materials at the same time, which is really cool. This is moderately quick, but the problem is that you're going to have some weak and brittle parts, especially between the Z layers. And so the Z direction is perpendicular to the print bed. And so in this picture, it's the vertical direction. Your surface finish quality is low, but there are some tricks that you can use to enhance your surface finish quality. And your size is going to be kind of based on your machine height and your base. So there's a lot of things where you can see they've 3D printed houses using kind of cement in the same type of configuration. And it's just based on how big your 3D printer is. Resolution in the XY plane, so in the plane of the print bed, is going to be dependent on your nozzle diameter. And in the Z direction, it's based on your mechanism to raise the nozzle. And so if you see here, there are vertical rails, and that's what allows you to raise your nozzle up. So one of the things that I want to talk about in particular, and I think let's remove the spotlight so I can show some objects in my video. Thanks. Yeah, so, or actually, yes, the spotlight, but not the slides, my bad. So this is support material. If you have a 3D printed object, you may not be able to just print only the object that you're 3D printing. Oftentimes you're going to need to print support material to hold it in place so that it doesn't deform, or so that the fused filament doesn't just like fall down onto your print bed when you're already this high up, right? So removing support material is probably one of the most labor intensive parts of post-processing your 3D prints, and it can be really, really difficult. So I'll tell you a little bit more later about how to plan for that, but I also wanted to show you a few different things that I've 3D printed using an FDM printer. So I've told you that the surface finish can be kind of poor, and that the machine makes these weak and brittle parts. But one of the things that my students have done recently, and I don't know if I have enough light to show you this, there we go. They've made a fully working 3D printed gearbox, and it's strong enough to run a quadrupedal robot. And the way that we've done that is by cleverly orienting the print in the Z direction so that we don't have a lot of stress to break apart the layers. Other things that I've 3D printed in the past include like these beetle heads, because we were trying to find out how the shape of different species of beetle heads corresponded to their fighting style. I've also printed out these snake fangs because I'm doing a project on snake fangs. You can see the hollow tube where the venom goes through. And one thing that's really nice about these FDM printers is that you can create hollow prints. So you're not wasting too much material by making this a totally solid print. There is some amount of hollowness on the inside. You can also 3D print things that are already articulated, just let them kind of loose by removing the supports. So there's a little articulated octopus, and then we've also got like a 3D printed sandworm that's kind of modular here. Okay, yeah, we can go back to the slides. I just wanted to take more time on FDM to talk about all of the different terms and introduce them to you. So yeah, we've got supports which are necessary for overhangs, and post-processing, you got to remove the supports to get a functional piece. There is a lot of other fancy stuff you can do, and talk to me about that afterwards. But we'll move on to the next type. Okay, Polyjet is one of the more expensive types of 3D printing. So if you're thinking about Polyjet printing, I would get together like a group of different professors to buy it together, like write a big grant to fund it, or ask your department, or create like a shared services center or something for this. It's going to be a lot of money. You can print multiple materials at the same time. You can print multiple colors. It's basically like you're taking this resin and you're spraying it in place, and then you're using light to cure it and to harden it in place. So it's really fast, and it has this beautifully high surface finish quality. But it can be pretty inefficient to do post-processing. Okay, we can move on to the next slide. So Sla Stereolithography is also really, really popular. And this is, we're getting into kind of these resin-based 3D printing. It's moderately priced. You can get a cheaper SLA 3D printer for around $3K, but you can also go up to the tens of thousands of dollars. There's a ton of different materials you can use. A lot of them are harder, but some of them are also flexible and elastic. And they come in lots of different colors. Some are durable. I even 3D print in high temperature resin. It's a medium speed. You get a pretty high surface finish quality, but you know, you've got to do a lot of post-processing. You've got to dissolve the excess resin in alcohol, and then you have to figure out what to do with all of this contaminated alcohol. You've got to cure it in UV light. One thing to note is that when you're thinking about the supports, you're actually printing things upside down. You're embedding this test bed or this print bed into the bath, and then you're using a laser at these different points to harden it, and then you're pulling it up. So your supports are going to look a little bit different when you're pulling your print up versus when you're supporting it, printing it above. With SLA, you cannot print hollow parts. They have to be solid, and we'll talk a little bit more about why later. The other thing about just resin printing in general is it can be a little bit wasteful because you have to fill an entire tank with resin, and you're never going to be able to use that all up. So there's always going to be a little bit of waste there. Okay, next slide. So DLP is another type of liquid resin-based 3D printing. It's a lot cheaper than SLA. And the way that it works is instead of having your test bed or your print bed, why do I keep saying test bed? Your print bed submerged in this resin and having a laser come along to harden that resin, you have an entire LCD screen and it hardens an entire layer at a time. So it's much faster than SLA, and you have a pretty high strength, but potentially a lower surface quality, but that may not be important for what you need. Again, it's going to be a gravity supporting upside down print. And the post-processing is going to be about the same as with SLA. Okay, next slide, please. Great. So we've got SLS, which is selective laser sintering. These are really expensive. So again, this is something that you might want to get a whole bunch of other people together to buy one of these. And you're going to need some specialized facilities, which I'll get into. For materials, nylon powder is kind of the main material, but you can also do direct metal laser sintering. So we've been 3D printing in titanium as well. Again, there's going to be some waste because the way it works is you lay down a layer of powder and then you take a laser and just harden all of the parts that you want to be hardened. And then you have another layer of powder. So because you are embedding this in powder the whole time, for nylon powder, you're not going to need any supports. It's just all supported by that unhardened powder. But the problem is because you have to heat up the entire set of nylon powder to just a couple of degrees within melting so that the laser, just by shining that light, will melt it. That powder is not 100% reusable. So you're going to have to mix it back in with some of the new powder so that it's not too poor of a quality, but it is somewhat recyclable. So there's a little bit of waste, but there's a lot of recycling that can happen too. This is really, really slow, and the surface finish is very grainy. Where did I put that? So you know that little, the octopus that I showed before, here's one, if we stop SlideShare, here's one made with the SLS printer that we have. It's still really functional, but the surface quality is actually pretty matte, and it's pretty grainy. Now, the other really terrible thing about SLS printing is that it's kind of like excavating in terms of post-processing. You're sort of like an archaeologist and you have to like dig out every single piece and brush it with like a toothbrush. I actually recommend dental tools if you're going to get this. It's a giant pain. And that's because this powder is a lung irritant and it's really bad for your health. And so if you're going to get an SLS printer, you definitely need to get proper ventilation, a lot of proper gear. It's also combustible and an explosion hazard. So you've got to make sure you have all of the safety precautions available. Okay, we can go back to the slides. Okay, great. Yeah, so next slide, please. Yeah, so I'll tell you a little bit more details about FDM printing, but these are all kind of general and very quick. All 3D printers are going to have a slicer software, and that's going to take this 3D object and then turn it into the individual layers that are going to be sent to your 3D printer. Usually your slicer is going to be able to generate support structures for overhangings if you need supports. they can also help you try a bunch of different orientations to reduce your supports and make cleaning a lot easier. So if you take this T-Rex head and say you had it horizontally, you would have a ton of supports inside of the mouth, and those are going to be much more difficult to clean out than if you have it vertically like this. For SLA printing in particular, you can't have hollow prints because you're just pulling that up from nothing, from just being on this surface of the liquid. And so if you pull up a hollow object, it's going to create a vacuum, and that's going to create a negative pressure that's going to damage your print as it's printing. And so your slicer software should be able to identify cupping, where you've got concave areas and you're in danger of this cupping artifact. For SLS, for powder, it's also really helpful to pack as many things as possible into that 3D space to minimize waste. So if you're trying to 3D print a ton of things for, say, a class, this might be a really great way to go. Next slide, please. Okay, so here are some trade-offs. And all of this can be controlled by slicer software. One of the biggest issues if you're working with 3D printing of any type is figuring out when people can schedule themselves in to use the 3D printer, because everyone's going to want to use it. And so you can reduce your print time by reducing quality, size, increasing simplicity, or reducing strength. And strength itself is something you might want to optimize as well. So strength increases with number of shell layers. If you look at the very bottom part of this slide with the, right there, with those two pieces, the one on the left has fewer shell layers. You can see the outline there is a lot thinner. And then the one on the right has many more shell layers. So that's going to take a lot longer to print, but it's going to be a lot stronger. On the top image, we have Infill. So this is a hollow object from the STL file, and the slicer automatically puts in a random infill. And as you go to the right, you increase the percentage of infill, and that increases the strength, but it also increases the print time. And then a higher Z resolution, so being able to lift your nozzle up or lift up your test or your print bed in much more fine detail can also increase the strength, but also increase your print time. Next slide, please. Multi-material printing is really, really cool. This is especially possible with Polyjet or FDM, and it's especially useful for making dissolvable support structures. So you don't have to go in and clean it all by hand. You can just put it into a bath of either water or alcohol or some kind of solvent. I highly recommend, if you want to do this, going to this link, because it takes you through all of the different ways that you can create dissolvable supports, all of the different technologies and the pros and cons of each. It's extremely thorough. I just want to caution you that the material used to create these dissolvable supports may swell in humidity, or it might break down prematurely if you're not storing it properly. So just make sure that you don't buy tons and tons of it and then don't use it for 3 years. That's not going to work out well. Okay, next slide. All right, for my last slide, I just want to let you know that Jennifer Lewis is a professor at Harvard who is incredible at figuring out new ways of 3D printing. One of the things she does is 3D print embedded in gel, and also she can 3D print directly in air really precisely. So I highly recommend going to that YouTube link and watching her talk. It's the future of 3D printing. So with that, I think I'm going to hand it over to Henry Astley.
All right, thanks very much, Talia. And I'm going to walk you through a project that we did in my lab to sort of show you how you can go from sort of start to finish on a single project, incorporating various aspects that Talia and Brooke and Em have talked about previously and that were in George's slides. This project was one of the thesis chapters of my student, Derek Jurastovsky, now Dr. Jurastovsky, who will be starting at Penn State University as a postdoc on Monday. So yeah, it's a really fun project. I really like talking about this one, which is basically trying to figure out the function of a particular anatomical feature of one of my favorite groups of animals, snakes. So snakes are, of course, very long and flexible organisms, in part due to the sheer number of vertebrae they have, often 150 to as many as 400 and more. And there has been some study of the morphology of these vertebrae and all the little processes and whatnot that you can see sticking out of this prairie rattlesnake vertebrae. And some of these are familiar to us, like the cotyle condyle. They have a ball and socket joint, the neural canal, the neural spine, pre and post zygapophyses that interlock. But they also have something special that you see. Next slide. So this is a special additional joint that snakes have called a zygosphene, and it has a corresponding cavity on the posterior surface of the vertebra called the zygantrum. It is always prominent and well developed in every snake, regardless of habitat, regardless of phylogeny. Every single one of them has a significant prominent zygosphene, and this feature is missing in all other vertebrates except for a few lineages that have something that is minimal and barely even counts, in my opinion. It's very small, very subtle. These have a much more prominent sort of role. So the big question is, if this is the universal feature of snake vertebrae, what's it for? And the traditional hypothesis advanced by Romer is the prevention of twisting. So in most of our vertebrae, twisting is limited by the pre and post zygapophyses, but if you're a long, skinny snake, maybe having this extra joint somehow helps. Next slide, please. So experimental manipulation is basically off the table for this. This is buried deep, deep in the vertebral column, inside of its own sort of articulation with the successive vertebrae, right next to the spinal canal. So manipulation is basically impossible and probably would be lethal. And even if you could manipulate it, you'd never be able to rule it apart from a sham surgery that was similarly invasive. And traditional comparative methods are not terribly useful either, because every single snake has it. So there's only one origin, and everything else that lacks it, even other limbless squamates, there's too many differences in the vertebrae to give an apples-apples comparison. So here you see, above you see a prairie rattlesnake vertebrae, below you see a legless lizard vertebrae from a paper by Cernansky et al. And the legless lizard lacks a zygosphene, but it's also got a different shaped condyle, cotyle joint, different angles for the zygosphene. You just can't make a good comparison with this, especially when you only have one origin for it. So how do we solve this? Next slide, please. So we adopted this idea from Kleintek et al., from their lovely paper on caecilians, where they refer to it as experimental morphology. This is the practice of manipulating the observed morphology in a living species into alternatives, using digital or mechanical methods and testing these alternatives. So they were faced with two skull types that you see in caecilians. One group had prominent fenestrae shown with the arrow, others, everything was all fused up and they lacked these fenestrae. And they were curious as to whether or not this had any effect on bone strain. So they, instead of having to deal with all the many other differences between species, they simply took some CT scans of the skulls, sealed over the fenestrae in some, removed bone to make fenestrae in others, and examined the effect on bone strain using finite element analysis. They unfortunately detected no effect, but it was a fantastic study in the potential of this method. So we decided to adopt it for ourselves to examine the zygosphene in snakes. So we've got our clear hypothesis. Now let's go on to methods. Oh no, yeah, please go ahead, sorry. So we use a similar approach, but use mechanical manipulation rather than entirely in the computer. So the first step, we need the morphology. As Brooke pointed out, CT scans are a fantastic way of doing this. We have a micro CT scanner on campus and a bunch of snakes in jars. So We got some scans from 4 different species across the snake phylogeny. So you can see the CT scan here. So go ahead and next slide, please. So here you see the vertebrae. We reconstructed them with using standard, I believe it was Slicer more for one of the other Slicer softwares to reconstruct STLs of the vertebrae and fix any of the various problems that come up using Mesh Mixer. So next slide, please. So the big thing we did was we digitally removed the zygosphene. So up top in green, you can see a corn snake vertebrae, and you can see the zygosphene prominently on the top in the dorsal view and on the right in the lateral view. And we basically artistically removed it using Mesh Lab. That's the software. Using Mesh Lab to just sort of remove polygons and then smooth over the holes. So we have something that is not biological, but it looks as if it could be. There's no weird spikes sticking out. It's smooth and sort of organic looking as best we can make it to make a fictional morphology. So now we've got our 2 test sort of configurations, normal and altered. So next slide, please. So next we used an FDM printer, as Sally was talking about, and scale was an important question. Snakes, even very, very large ones, tend to have very small vertebrae. And so we made really enormous vertebrae. You'll see in the next slide just how big they are, but they were printed out at 14 times size. So these are truly enormous vertebrae so that we could manipulate them more easily and track their movement. So we used a fused filament printer. You can see the support material here, printing in this particular orientation, because one thing that we ran into was that if any surface on the lower surface of the print would be more rough due to this support material. No matter how careful you are in removing it, there's always some roughness there. So next slide, please. So we then articulated 2 successive vertebrae from our CT scans. Some of them had a zygosphene, some of them didn't, and moved them through a full range of motion as much as we could get them to, while keeping the cotyle condyle joint articulated and using motion capture to track the position of the anterior and the posterior vertebrae using our cameras. So next slide, please. And finally, we had to make sure that what we were doing was biologically relevant. Our tests don't mean a lot if all of our differences that we find are beyond the range of what snakes ever do. So we had ex vivo bending tests where freshly euthanized animals were maximally bent laterally, dorsally, and ventrally to get a sort of range of motion and see how far is vertebra and joint-on-joint contact versus what's limited by soft connective tissues. So next slide, please. This is an example of our data from an intact or unaltered vertebrae. So this is from a boa constrictor. And so the black and the graph here in A on the left is pitch versus yaw. So you can see the snake can go about 20 degrees left and right, and about 10 degrees up and down relative to a neutral posture. And the red lines denote the range of motion from the ex vivo tests of a freshly euthanized animal. So the B and C panels are essentially looking at the roll axis. So B is essentially A, looking at it from the side. C is basically looking at A from the top view. And in all of these cases, roll is minimal, less than what we would get from just a static object and the random jitter that you get from any motion capture camera. So minimal roll. Next slide, please. When you remove the zygosphene, things got really different. We expanded the range of motion in pitch and yaw, particularly in these sort of high pitch and ventral deflection areas that are highlighted in blue. And they're highlighted in blue because these areas are also the areas where roll suddenly became enormous. So the roll, even in the altered zygosphene-less case, were minimal until you got to this particular configuration of very low pitch and very high yaw, either to the left or to the right. Because what was happening is the pre and post zygapophyses were disarticulating on one side. And as a result, it could freely spin in one direction, which is why you see what you see in panel F, where it can roll, but only in one direction. And then it goes to the other side and it can roll in the other direction. Next slide, please. So the cool thing is what we found was actually a completely different function, or a different function than was originally hypothesized. It was originally supposed to purely be for prevention of roll, but across most of the range of motion, the pre and post zygapophyses are more than sufficient for that. Instead, it seems to function as a bony limit, as a blocker to prevent the vertebrae from actually getting to these trouble areas, which would have allowed the disarticulation of these other joints and therefore preventing roll. So it's not preventing roll directly, it's from preventing you from getting to a point where roll becomes a problem. It also might be useful for providing additional mechanical force at high ranges of motion under high load, so constriction. Most of these species, not the rattlesnake, but all the others are constrictors. And so this joint might have a function in that as well. You want to be able to tighten your body as much as you can without having to worry about accidentally disarticulating your own vertebral column. So yeah, next slide. All right, with that, we'll wrap up, and next workshop will be next week at 11 a.m. Eastern Time, 8 a.m. Pacific Time, and we'll talk about actuating your physical models. So that I'll turn it back to Em and everyone else, and thanks everyone for listening.
Great, thanks everybody. I think that's it. I'm excited for next week. I'm going to learn some things from you, Henry. And thanks a lot to Monica for making this workshop and the series happen. It's been really great.
We can hopefully you can stick around for a while in case anybody in the audience has questions. You can go ahead and post questions in the chat or in the Q&A box, or you can just raise your hand and we can unmute you to ask your question. If there are no initial questions, I might start out with one. It's kind of a big picture question. For those of us who might be thinking about how to approach a specific question, I can see like many benefits of physical models, right? Because they're like, you know, hands-on, and you get sort of like a real tangible kind of perspective on things. But I wonder if you might be able to elaborate a little bit on trade-offs between approaching a problem using simulation versus physical models. Could we answer the same questions, or what do you think the trade-offs are there, like the advantages of physical models over simulation in particular?
I can start. I think, not that I have huge experience in either of those things, but what I do have experience with is that in simulation, I think sometimes it is really hard to capture some of the intricacies of fluid interactions in particular. And so the equations start to get so complicated so quickly that you can't capture all the physical principles. And so you can throw away all those major equations if you just build a model and tweak it in real life. So I think that's,
you don't have to have equations for the physics of the system to do the tests. Yeah.
Avoid math.
Yeah.
Well, that seems to make it a lot more approachable for a lot of biologists then, right?
Yes.
I mean,
Like myself,
I got into
right?
this area actually when I was in Dan Goldman's lab, when I was a postdoc, because he works on sand, and there are no equations that govern it. If
There are
you
no
want
good
to
equations
simulate
for sand dynamics, too complicated.
you have to simulate every particle, which rapidly becomes infeasible. But you can just make a robot do what you want, and nature does the computations.
Mm-hmm.
Yeah, your simulation is only as good as your physics engine. And when you create a physical model and you actuate it in a real world environment, you may actually make discoveries that you didn't know or you weren't looking for in the simulation, and then you can use that information to update your model in simulation. So I think it's worthwhile to do both and have them inform each other.
That's great. We do have one question in the Q&A. How did you do motion control? Any recommendations for dipping a toe in that area?
Stay tuned for next week.
Yeah.
Oh, okay. So you're going to cover that next week. We have another question. Can you see the questions?
Yeah. So the question is, linking the physical model to the biological model. How to persuade other people or rigorously verify that the robotic model is actually reliable for testing biological parameters? That's a great question. Yeah, I think sometimes it's tricky. And I think that making one of those performance landscapes can be helpful, because if you use your robot or your model in a wide range of areas, and there is overlap with what we know what biological systems do, that's pretty compelling. And so I think that's one way I would think about putting rigor into the model, is that to cross test it with biological systems, what you can get a hold of. I don't know if anyone else wants to add on that.
Yeah, I would. I just say absolutely. That's that validation process I tried to talk about a little bit. And if you're struggling with this, say with reviewers or something, a big thing to do is to take note of the fact that, you do know that there's certain material properties that exist in your robot, in the organism, and that you've modeled them in the robot in this way to be similar. You might talk about how you have the same physical environment in terms of Reynolds numbers if it's an aquatic environment or something. And then if you can match that parameter space, like we showed with matching the kinematics of the organism in the model, and then getting similar force readings, you know that now you have a good proxy for the organism itself. So I think if you can kind of check off all those boxes, that you're in a good place, that you have a well-validated model.
There's a saying that I've heard a million times, and I'm sure we've all heard it: all models are wrong, but some models are useful. So no model will ever be a perfect snake, a perfect fish, or a perfect jerboa, a perfect mud skipper. But if it's testing the hypothesis that you want, if it's similar in the ways that you want, then how much does it matter that it's different?
Yeah, and I think that it's always good to make sure that you list all of your assumptions, because they're always going to be assumptions, right? You're never going to make a perfect biological mimic, but as long as you're aware of what you are assuming to simplify your model, and you're making the community aware of what you're simplifying, I think that can be really convincing.
All right. Do we have any other questions from the audience, or questions for each other here among the panelists?
I'm going to have questions next week.
Okay, well, I guess we'll wrap up for today then. Thanks everybody for tuning in, and I hope you'll join us next week to learn more about this really great topic. And thanks to all the panelists for your talks. I learned a lot. It's really useful.
Well, that was fun. Thank you, Monica.
Yeah, thanks for organizing.
All right.
Thank you.
Thanks,
Yeah.
everyone.
All right. Signing off for now.
Bye.
Bye. Thank you.
See you.